Hi, my name is Kat Greenburg, and I have been working in Dr. Marcus Babst Lab in the Cell and Genomic Science Center at the University of Utah. The Babst Lab focuses on up and down regulation of cell membrane transport proteins and their response to environmental stress. I specifically have been working with leucine, which is hypothesized to be one of the most important mediators of metabolism in Saccharomyces cerevisiae, or baker's yeast. Um, Leucine itself is taken into the mitochondria where it regulates metabolism, just all of metabolism. And it's hypothesized that when leucine is not present or the cell is unable to make leucine from glucose, the cell will trigger cell death. When exploring cell starvation of glucose such that it can't make leucine, we hypothesized that leucine would be taken in higher amounts from the cytosol. But we found that under starvation conditions, the amount of leucine taken in versus the amount of leucine made is no different. The ratio is the same. And that is shown outlined in the data in the center of the graph. The top center shows starvation conditions, not starvation conditions, and a 1:3 ratio of leucine taken in versus leucine produced. That data was acquired through liquid chromatography mass spectrometry, which is the graphs shown in the center. And when doing this, we found that we were able to quantify the amount of leucine present, but we are not able to distinguish between leucine and isoleucine, which have the same molecular mass as they are isomers of one another. While we tried to decide how to differentiate between leucine and isoleucine, we started to explore mitochondrial degradation when you knock out the general amino acid permease, which just allows amino acids to leak out of the cell if they are accumulating to cytotoxic levels. The bottom fluorescence microscopy shows mitochondrial degradation when we knock out the general amino acid permease, and then healthy mitochondrial growth on the right. So left, it shows the mitochondrial degradation, and right, it shows healthy mitochondrial growth. Further exploration is required to figure out if leucine is the amino acid that's most important in this mitochondrial degradation process. And various knockout strains will be created and explored under the same conditions as we have shown and demonstrated here. Are there any questions regarding this research? How do we plan to separate isoleucine from leucine? That's a good question. This will have to be a collaborative effort of various labs who have different lab equipment. So high pressure liquid chromatography is a method that we can use to separate isoleucine from leucine in the sample. We can then take the leucine samples and do liquid chromatography mass spectrometry, and that will tell us how much of the leucine is present in the media. So that's how we plan to separate isoleucine and leucine in the future. What are the various starvation conditions mentioned? Yes, so these baker's yeast cells need about 2% glucose to grow efficiently. As shown in the chart, we have 1.4 optical density versus a 0.8 optical density when you provide the cells 0.2% glucose. So the 0.2% glucose doesn't immediately kill the cells. It allows them to grow to some degree, but then they can't grow past that optical density of 0.8. So they are borderline starving. We also ran experiments using 2-deoxyglucose, which is a glucose variant that when taken into the cell cannot be metabolized. So the cell will accumulate 2-deoxyglucose, but it can't break it down. And it won't intake any more glucose because it believes that it has enough glucose, which is actually the 2-deoxyglucose. These are great questions, thank you. Well, if nobody has any more questions, I would like to thank you all for your time in stopping by this presentation. You can follow the work online at Marcus Babst's website, which is on the University of Utah website. I would like to thank Dr. Babst for all of his mentorship and help in this experiment. I would also like to help Lincoln Gay. He is a graduate student in the Babst Lab, and he has helped me immensely with methodology and experimental design. I would like to thank the Undergraduate Office of Research program. They have funded my research and allowed me to do what I did in this research project. And just everybody who supported me in my research and taken the time to stop by today. Thank you.